Welcome to the CBIA BizCast. I'm your host, Ali Warshavsky, and today on our podcast, we are speaking with Dr. Charles Lee of Jackson Labs in Farmington. They have been extremely busy during the pandemic, as you can imagine, and now they are helping determine which cases in Connecticut are which different variant. So I'd like to welcome Dr. Lee to the podcast.
Hi, Ali. It's nice to be here.
It's great to have you on, and we really appreciate you taking some time, which is probably a very busy time right now as those COVID rates have started to go up. Let's go back, though, to March of 2020. How has your role changed since the coronavirus was found in the United States?
Yeah, so as for everybody, I'm sure everybody has their stories of where they were and what they were doing as the pandemic started. For us, it started really with the fact that early on in the pandemic, we realized that this virus was sweeping through the globe. And we also realized that the recommendations early on were that anyone that had a suspicion of having COVID-19 infection, they were to get the samples sent to the CDC lab in Atlanta. And it's pretty obvious that with something like this, it's send the samples to one lab, even the CDC, is just not going to be enough. And so we reached out actually to the CDC, both the federal, state level CDCs, and there was a protocol that they were following. And so finally, we decided to go straight to the governor, Governor Lamont, and we asked him, look, we have a clinical diagnostic lab at the Jackson Laboratory here in Connecticut, and we could easily set up the COVID-19 test. And would you like our help on this? And so Governor Lamont, it was amazing to see him. He knew what he wanted, and he said, absolutely, we need all the help we can get. And so we started we started preparing for the test, and it took us about 3 weeks to get the test all up and running. Very few clinical diagnostic tests have been set up in 3 weeks from start to implementation. And on March 23rd, we started to launch the test at about 50 tests per day. And at the peak of the pandemic, we had ramped up the services so that we're actually running about 10,000 tests per day for the state of Connecticut. So it's a huge increase. And it's been, it's been really satisfying to be able to be at service for the citizens of Connecticut like this.
And you said it's very rare for it to take 3 weeks to set something like that up. Is there anything in your career, do you remember, that was anything similar to this, where there was a very quick turnaround?
There's nothing. In fact, before coming to Connecticut, I was at Brigham Women's Hospital as a certified laboratory diagnostician. Brigham Women's Hospital is a Harvard affiliated hospital. So I've been among the best. I've seen the best at action, and there is nothing that compares to that. This was all hands on deck, and I'm so very proud of the people that put this together right here in the state.
And when you said you were processing around 10,000 tests, what did that mean for employees? Like, how many do you need to process that many? Are they working all hours of the day and night?
Yes. So initially, of course, we started off with one shift of 8 to 9 hours, and probably about 17 people that were working in the CLIA lab. And as we ramped up, there were two levels of ramping up. So one is, of course, they're increasing, as you are implying, the number of people working in the CLIA lab. So at the peak, we actually went from about 17 individuals in the CLIA lab to about 78 people. And so you can imagine just the aspects of trying to hire people and train them in this process is a Herculean effort in itself. But then on top of it, that's not enough. You actually have to implement a lot of automation robots, and even having extra tissue culture hoods, etc. We ended up purchasing well over $11 million additional equipment to get this automated to the level that we could do 10,000 tests per day. So both of those in combination, the increase of the staff and training them, as well as purchasing and implementing this automation was what made it all possible.
Wow, yeah, that is a lot to be going on during a pandemic where no one really knows what's going on, what's going to happen next. And even though a lot of us, we keep saying, oh, during the pandemic, unfortunately, we're still in it. And you guys know that better than anyone, because now you're helping the state determine how many positive tests are which type of variant. How does this happen? And I believe I read it's genomic sequencing. Hopefully I pronounced that right. But
Yep.
For us non-scientists, what exactly is that? How does it work?
Yeah, so you're absolutely right. And actually, so the way the testing works is the actual clinical diagnostic test is called a PCR test. So what the PCR test actually does is we've identified, the test identifies 3 regions on the viral genome, which it targets and then it amplifies, or it tries to amplify. So for example, if you have a swab that's taken for an individual, you want to know if they have SARS-CoV-2 virus. That swab is then mixed with the solution. If there's any virus that goes into the solution, and then you're asking the question, is there any virus in that solution? And the way you identify it is you target 3 regions of the viral genome and you see if you can amplify it. So if the virus is there, those 3 target regions will amplify, and you'll get increased, we call it fluorescence, an increased signal. If the virus is not there, we won't get any increase in the signal, and it's a negative result. So, so that's how you do a typical PCR-based SARS-CoV-2 test. Now, in some cases, so when you want to actually do to determine what the variant is, you actually have to understand not is the sequence is present, those 3 areas you're targeting, but you actually need to understand what is the actual sequence of the entire virus, and then what changes are there along that sequence compared to the original strain of SARS-CoV-2. So you're absolutely right. We essentially sequence the entire length of the virus is about 30,000 bases. So we sequence that entire 30,000 bases, and then we look for what are the changes along all 30,000 bases. And of course, that by doing that by hand is almost an impossible task. We have computers that help us do that quickly. And then based on what changes are in that virus that we are sequencing, we try to identify a signature that tells us, is this the alpha variant? Is this a beta variant, delta variant, etc.? And so that's what we do here at the Jackson Laboratory. So in addition to the PCR testing to determine whether the sample has virus or not, we do follow-up sequencing where we tell you what's the exact variant. I hope that makes sense.
Yeah, so it's obviously, you said, done by a computer, and I think most people would assume, like what you said, you get tested and then it's positive. Then if it's positive, it goes right to this computer or robotics that analyzes the way it's made up. It notices that it's made up the way the Delta variant is typically made up, so you therefore have the Delta variant. When did labs decide to start, or I guess not, you probably didn't decide, but maybe the CDC, to run tests to see if there was any genetic changes or mutations, I should say, to the virus? Because for so long it was just COVID. How did they even identify that there was a different type of variant?
Right. So Ali, let's just make sure I've got the concept across that the actual DNA sequencing is actually another test above and beyond the PCR test. So once you've done the
Yes.
PCR test, you actually have to run the DNA sequence and then analyze the sequence. So,
Okay.
but then to your question now as to when did we realize we needed to do this? I mean, we realized that we needed to do this early on because biologists, infectious disease experts, they know that viruses mutate, they evolve.
Mhm.
And they know that as viruses evolve, have different properties in terms of infectivity, in terms of severity of the disease. So early on, it was really important to start the sequencing. Now, this actually may be a good time to show you one of these slides that I prepared. In this particular slide, it actually shows, if you look at the slide that says SARS-CoV-2 genome sequencing, you can see early on during the pandemic, the different colors of waves that you see there is actually the different variants based on sequencing data. So that there was a level of sequencing that was happening early on in the pandemic. And for the most part, you can see the blue wave is the alpha that represents the alpha variant. And so that was a predominant variant early on in the pandemic. Then you can see on the right side later on in the pandemic, including up to now, the vast majority of the viruses that are being sequenced are what we refer to as a Delta variant. And there's a lot. It's really taken over all of the viruses that are being sequenced right now. And so being able to identify these waves and then watch how the patients that are infected by each of these different variants respond to treatments or how many of them are being hospitalized, how many of them have been vaccinated versus not vaccinated. I mean, this is all important information. And without knowing what that variant is, it's hard for us to prepare for next steps.
Interesting. So this has been done with something like the flu before. This is not new to the to COVID-19. You've looked at this when other viruses, again, commonly the flu, have gone around every winter.
That's exactly right. So every winter, primarily it's the CDC that actually collects a portion of the samples, sequences, does the genome sequencing, finds out where mutations are, what are the different variants that we're seeing. And this actually helps them to, on one hand, it will, through epidemiological studies, it will help them understand why a certain, the flu vaccine that they used was either very effective or not as effective in a given season, prepare them for the next season. And so the more information on this, the better. And traditionally, the CDC has been doing this. With SARS-CoV-2, because of the nature of the disease, it's sort of, again, all hands on deck. Let's all contribute as we can to get as much information possible to combat this pandemic.
And speaking of information, what are you finding at Jackson Labs? I think as of right now, which is the 13th when we're speaking, there's been several of the new variant found in the state, but not too many yet. Of course, by the time this is released, that could change. But from what you guys have been studying, what are you finding? Is it still predominantly the first coronavirus mutation that's here, the Delta, the Omicron? What are you guys seeing?
Yeah, so what we've been seeing is so to give over the past month, we have had 302 positives that have come through our clinical diagnostic lab, and we've actually sequenced each and every one of those. We're just to put things in perspective, Ali, the United States is aiming to see approximately 5% of all variants on average. just to give us a landscape, because it's a lot to do every one of them. The state of Connecticut has actually gone further than that. And the state of Connecticut's goal is to actually sequence about 20 to 25% of all the variants in the state. The places that are contributing to our Quest Diagnostics is doing some sequencing. Nate Grauber at Yale University is doing a considerable amount. Of course, the Connecticut State Lab and the Medicine Laboratory. And we've made it a policy here at Jax. We're actually just sequencing all of the variants, all the positives that we get. And so what we're seeing up to this point is that everything, pretty much 99% of all the variants we're sequencing are the Delta variant. Now, having said that, I did not prepare this for this podcast, but we now actually have a suspicious Omicron variant that we believe we've identified. We're trying to confirm it now. And your listeners may be asking, how do you determine a suspicious variant? And the reason why is, remember I told you earlier on that for the PCR assay, we actually target 3 regions of the genome. And what happens is 2 or usually all 3 of those regions, if there's a virus, they all go up and you get an increased fluorescence signal. And sometimes one of the regions might not work, but for the most part, all 3 of those regions go up if there's a virus present. Now, interestingly, one of the mutations in the Omicron variant actually prevents one of those regions in the PCR assay to go up properly. And so it gives us an indication when we have the other 2 going up, but not that third, that this may be a potential Omicron variant. And so now we're doing the whole genome sequencing, and that'll tell us conclusively if that's the case.
Okay, so what you're saying is from what you guys have been searching through and analyzing, your specific lab hasn't had any, but you might think you now have one that you are analyzing that is of that variant, because the ones that have come out throughout Connecticut have been through other labs, correct? Is that what you're saying?
That's correct. Yeah, and my understanding is actually it's primarily the state lab that has been picking up the current variants. We have, last that I saw, 11 confirmed cases of Omicron in the state.
This is something I've been wondering ever since I started testing for Delta, but how do you know, or how do you guys determine which ones go through that testing to see if it's a variant? Because, you get swabbed, you usually are just told that you're positive. How are you, I say lucky one, but really you're unlucky if you catch COVID, to then be contacted and them tell you, oh, also we found out that you are the lucky carrier of this variant. How is it determined whose test gets further analyzed?
Yeah, that's a great question. So for most of the labs in the country, they would randomly just identify 5%, right? So if for every 100 positives, they might just they'll randomly choose about 5 of those, run them through the sequencer and see what the variant is. Here in the state of Connecticut, as I was saying, we're trying to do better than the US average. And so as a state, we're actually collectively trying to sequence between 20 and 25% of all the positives. So we have a more thorough understanding of the variants. At the Jackson Laboratory, we're just decided we're going to sequence all the positives. So anything that comes to us, might ask, well, what are the samples that come to the Jackson Laboratory? Right now, we're primarily doing the tests that are coming in from the nursing homes, about 120 nursing homes across the state, extended living places. And so among those samples, any positive that we get, sequencing them. So
interesting.
So we sequence them all.
Are you obligated to tell someone if they their variant is different, or just you guys do that just as a courtesy?
So all of the information that we get actually goes to the state
Okay,
directly, and it's part of their surveillance program. So
gotcha.
when Yale does their sequencing, Quest does the sequencing, we don't actually give that information back to, so the state will collect that information and then determine next steps. It's
So
coordinated.
interesting. Oh, thank you for explaining that. I think a lot of people were wondering, like, if my test, how do I know if I'm one of the tests that gets the further research? So I just want to clarify because I think your audio might have gotten cut off a little bit in the beginning. The national standard is 10%, Connecticut's is 20.
The national average is 5%.
Average 5%. Okay, wow.
5%.
So we're way above what is required in Connecticut, which is always nice to see.
Well, actually, Ali, if I may, let me just also show you this one slide, because I think it's important for us not to rest on our laurels, but just to be reminded of how good we're doing as a state. So in this particular, you can actually see that on the bottom, you can see the number of cases of COVID throughout the US and the different waves that are happening. And on the top graph, you can actually see what we're seeing in Connecticut. And you can see that the peaks are actually much more lower than the overall US. And I think that's a credit to what we're implementing in the state. So being very aggressive with the masking, vaccinations, testing, etc. So I wanted to put this up here just so that we can feel very proud of what we're doing and hopefully feel safe about the measures that are being placed for our state.
Thank you for sharing that. I do hope it helps ease some worries. It's been a weird time as those rates climb because of winter. And, you guys have another location in Bar Harbor, Maine. There's a lot of other research you're doing surrounding COVID and testing and sequencing. Can you tell us a little bit about some other projects that Jackson Labs is working on?
Yeah, so we actually have additional sequencing of the virus, first of all, that's happening for the residents of Maine. That's actually being led by one of our investigators, Ryan Toohey, and he's working very closely with the Maine CDC on collecting the information and taking next on that. But in addition to that, I'm not sure if everyone will realize that the Jackson Laboratory has been founded on more than 90 years of using the mouse as a model for human disease. And we're very proud of being really the forefront investigators in this area. And so capitalizing on that strength, just give you some examples. So Kat Lutz and her team have actually, right from the beginning of the pandemic, they've actually been the group that has been growing specialized genetically engineered mice that are specifically made to be used for vaccine research, to see the efficacy of vaccine research. So their team is constantly busy at providing hundreds of investigators in, I believe, 28 different countries in the world for that kind of research. In addition, one of the things that we all realize is the same variant of SARS-2, if it infects 5 different people, those 5 different individuals, let's say even if they were the same age, they can respond quite differently. Someone could be hospitalized, another person could get the sniffles, another person may not feel anything at all. And what is causing that variability? And so we believe, a lot of geneticists around the world actually believe that part of the answer lies in the differences in our genetics from one person to another, and what differences may protect one individual and not another individual from getting a severe infection, severe infection. So we try to model that in mice. So the same thing, we take the specialized mice that I talked about earlier, and we create those mice in different genetic backgrounds, and then infect those mice with the SARS-CoV-2 virus. And sure enough, we actually do already, the preliminary data shows some of the mice succumb to the disease very quickly, others have very little effect. And that could be a really powerful tool for trying to dissect what parts of the human genome could be leading to increased susceptibility or response to the virus. That's been led by Nadia Rosenthal and her team there.
It'll be so interesting to learn more about that, because I know that's seems to be one of the most confusing things about this virus is how different and how severe someone in the same family member could have it than you do. So we will look forward to talking to you more about that in the future when you guys release some findings. My last question for you is, how is the research being done by Jackson Labs helping to better understand this virus and then eventually curb the infection rates? You kind of just touched on it right there by the research of better understanding why it affects some people differently. But are there any other ways that you feel the lab is really helping to try to curb this and end this eventually?
Yeah, so so our investigators, of course, when the pandemic started, with their different specialties and their different knowledge bases, many of our investigators turned their attention to the SARS-CoV-2 virus. What could they do to help us further understand more about the virus and the pandemic? And so we actually have now several projects that have started that have actually gained funding from the NIH to pursue them. Just to give you an example, one of those projects is led by Daria Unutmas, who is an immunologist in the Connecticut campus here. And he's working very closely with the dean of the medical school at UConn Health, Bruce Leong. And they actually are looking at about 300 healthcare workers that looking at their antibody levels through getting the first shot, second shot, the booster, and how well does a sample of their blood neutralize the SARS-CoV-2. And one of the things they found, consistent with other studies that are going around in the world, is that indeed when between 3 to 6 months, the amount of antibodies that we have, even after the second shot, really wanes quite a bit across the majority of these individuals. And so we know that fighting the infection is not just about antibodies, it's also about the T cell repertoire and how they function. But at least one component, there's evidence that suggests from patients, actually it's healthcare workers here in the greater Hartford area, that have donated their blood for this research, basically shows that yes, between 3 to 6 months after your second shot, the number of functional antibodies that can neutralize this virus drops considerably. And so it reinforces the idea that the booster shots are really going to be important in the next phase of this fight against the pandemic.
Yes, it's very timely that this will be released, as many people are being urged to get their booster shot. Maybe hearing that will help them make their decision. Dr. Lee, thank you so much for coming on the BizCast. Is there anything you think we should know about what's being done at Jackson Labs that you didn't have a chance to mention, or about the coronavirus itself?
I mean, as I mentioned, we are we and researchers and medical staff around the world have been sequencing the virus from quite early on. And one of the reasons we'd like to do this is we want to understand the evolution of the virus itself. So this is actually a slide which you might find interesting. So on the left side here is sort of when the pandemic started. I don't know can you see this? And you can see the different variants that have arisen throughout the globe. On the very top, you can see this is the Delta variant, right? And that's the variant that's predominantly across in people that are being infected right now throughout the world. Now, we always thought that if a new variant arose, which was the Omicron variant, we always just thought that in fact it may be evolving from the Delta variant, because the Delta variant is all over the place. And there's more chances for it to evolve and create a new variant. turns out that the sequencing data doesn't agree with that hypothesis. You can see the Omicron variant down here. It's about 2/3 of the way down in red. And the sequencing of that Omicron variant actually suggests that it actually originated quite earlier on in the pandemic, did not come from the Delta variant. And so there's some speculations that it could have been a variant in, for example, a person with a very long-term infection. And during that infection, it changed and it changed and changed, accumulated all these mutations to the point where it now became something that could spread very rapidly. So having that kind of information alone is really important. That's why scientists really feel that gathering as much data as possible so we're the most informed is the best way to fight this pandemic.
That's very interesting to learn, because I think we all assume that every variant, the mutation comes from the last one. And you're right, like this graph looking at it looks like, well, as you said, it didn't. So
Yep.
Interesting to know. Well, Dr. Lee, thank you so much for joining us. I know I learned a lot. I'm hoping our listeners did as well. And thank you for listening to this week's BizCast. You can listen and subscribe to our other BizCasts on Apple or YouTube, SoundCloud as well. And for more episodes, you can visit CBIA.com. Thank you, Dr. Lee.
Thank you very much, Ali. Appreciate it.